Welcome to the first lesson of the first topic for Psychology General Senior Syllabus. Now, this is the old syllabus, which started in 2019, so grade 12s of this year, 2025, will still be following this guideline. For topic 1, localization of function in the brain, this is how the syllabus is laid out. The subject matter is the content that will be examined at the end of the year, with all the other information being guidance for teachers that pertains to the other assessments like the student experiment. Now, what I have done to help you construct your notes is I've taken the syllabus and put this topic into a terminology list. You will notice that the terms are indicated as bold or underlined or italicized. I will explain the purpose of that in a moment. Here are the diagrams that go with this topic. Both of these sheets are available in my coffee shop. This notebook I designed specifically with the purpose of using for notes in the senior syllabus. You should also have some sort of colored markers or highlighters. Those will help you to distinguish the use of the different terms as we go through the topic and where they will apply. Obviously, a good pen or a whole bunch of them, some sticky notes, some random stickers if you want to make your book pretty, some markers, anything else that is going to help make it colorful and nice. And because we're all human, probably have some of this correction tape. You could also light a candle whilst you are busy, preferably one that smells really nice. All of these things together is going to help to get your brain in the right place. Now, this notebook, as I've said, has been designed to have an index specifically aimed at the contents of the general subjects. In this case, we've got 3 different sections, which is why the terminology has been bolded or underlined or italicized. That is how we're going to break up this topic into 3 chunks that is much more manageable. I have listed them there and indicated the page where it starts. So you'll notice how the Cornell method lays out your content as terms, the main facts, and a summary. That is how we will be doing all of the notes, and because you know there are 3 sections, you can pre-plan the 3 pages that you are going to need for terms, main facts, and summaries. You don't need to use stickers like I have. You can obviously hand write those divisions. So then, for the terms, this first section that has been bolded, central nervous system, peripheral, somatic, autonomic, spinal reflex, monosynaptic reflex, and polysynaptic reflex, those will be the ones that has the main facts covered in this section. For the central nervous system, you will notice that first diagram that I have grabbed, even though it's printed in black and white, I have used my highlighters to match up the relevant colors as well. So your CNS, central nervous system, which you will understand, is the brain and spinal cord. PNS, everything else, divided into that red and the green, the somatic and autonomic systems. So the somatic being your body, so that's everything that you can control. Autonomic being your involuntary stuff, so heart rate, blood pressure, etc. Then we go to the spinal reflex. So that is the second diagram. Again, I have colorized it and added arrows and labels as necessary for your sensory neurons and motor neuron. That obviously is all grade 11 work. So the colors there are nice, transferable, and they will again stand out and match. So your spinal reflex, sending motor commands, sensory information. The direction is important from brain to body or from body to brain. Then we get to those 2 types of synaptic connections. So monosynaptic, mono being 1, and a polysynaptic being many. It's good to remember that monosynaptic is that knee-jerk reflex. It is something that you cannot really control. And then your polysynaptic is pretty much everything else. So double checking on the syllabus that you have covered the parts that you were supposed to. Recall cannot be directly asked in an exam, but it's assuming that you have that prior knowledge. So everything from grade 11 and science classes before that. So we have covered central nervous system, we have covered peripheral, the two types of the peripheral, somatic and autonomic. So this topic is good to go. Then getting to the spinal cord part, referencing the spinal reflex. Obviously your spinal cord is represented in your central nervous system, so that's covered. And the spinal reflex, we have done the two directions, from the outside towards the spine and then from the spine outwards. And we've also covered the two types of connections that can exist. So all of that is covered in that second section. This is a nice short topic, so we can then reach the summary section. The describing part can be something that is asked directly in the exams. In the next video, you will see I will go through some of the past exam questions that relate to this entire topic, and all the rest of those bullet points will be following on the next pages. So now for the summary. You can see there again I have transferred all the matching colors, because that is how your brain is going to match up and remember the things the best. Really nice and short and sweet, the basics of everything that you have covered there. Always remember to make sure that you have at least one kind of an example. So an involuntary action like your heart rate. It's especially good to keep in mind things that might be coming up in the topic to link to those things specifically. Then we get to the next set of terms. Now you will notice, obviously, this is a longer list. So you can anticipate perhaps using more than the single page you have set aside. In this case, I will just be using the one page, but I will show you how we can extend it. You will simply, in the space for your terminology, rewrite all of those. They tend to be in the order that they are covered in the syllabus document. They're not alphabetical or in order of importance or anything like that. You can double check again that you have them all, block them off nicely to isolate them in a little corner for you. It makes using this for the creation of flashcards also easier if that is the way you want to go. So checking on the syllabus document what we have to do. Again, the cognitive verb of recognize is assuming prior knowledge. This time we have to make sure that we are covering the functions of those areas as well. So one or two examples, maybe more if you can recall from previous years. Then the two brain diagrams. Obviously, we are starting with areas on the outside and not on the inside. So we won't be using the internal view, we will be using the external view to identify frontal, occipital, etc. So you can cut that out however you wish. Make sure it stays nice and in position, remembering that your placement is important, to leave some space above it to write about the cerebral cortex, the parietal lobe, etc. So there's that first terminology. Choose your highlighting color, mark it, and get the basics written down. In this case, the cerebral cortex is simply making it clear that it is the entire large brain section, what we generally refer to as the brain or the gray matter, that we will be talking about. So usually we would just say the cerebrum. Color it to make those connections. And on your diagram, because these questions are very often diagram based in the exams, you can indicate it like that. Do not go around your cerebellum, it's a separate part. So frontal lobe, again, here the specifics of the coloring is not important, as long as you can obviously remember that it is on the front part of that cerebral cortex that you are focused. Remembering that we need to pay attention to the functions of these discrete areas. So that is what we will be listing next. Again, I have chosen things which I know will connect to future topics. So voluntary movements will connect to our Parkinson's, for example. Personality is something that was related to the grade 11 work, so they assume that knowledge. Problem solving together with your higher executive is all going to come into play when we talk about the topics of learning and memory. Now, the next one on the list they gave us was the occipital lobe. Now, it is not obviously the lobe that is next in line if you're going around the brain. It is that area in the back. And you'll notice how I've left space just in case I needed that extra space to talk about the parietal lobe. So anticipating your layout as you go around is always very important. Again, linking the specific function to things that are coming up. We know vision is going to be a very big chapter, so we focus on those elements. We separate color, form, and motion, again, just because that is how they will be relevant in the topic. Next up, we will have a look at the parietal lobe. This time, you'll color in that top section. And focusing on the functions for that. So we know the parietal lobe is very much in charge of many of your sensory perceptions. So that can be a reminder to you that it's all of your senses, so tasting, hearing, touch. And you'll notice how I have shortened the words. Instead of saying hearing or smelling, I have changed them shorter to just make sure they fit in. This last function being the primary somatosensory area. Remembering somato means a body, so that just tells you that is where all of the senses experienced by your body is going to come to be interpreted and integrated into your understanding of what is happening. And lastly, for this external view, we have the temporal lobe, leaving space because the cerebellum on our list is coming up, so we need to make sure that we can talk about that. So the temporal lobe is going to be processing auditory information, again linking it to our prior knowledge and to topics that will be coming up. So we know we're going to be dealing with memory and emotion. And then obviously if it does auditory processing, it will process language as well. Next, we will do those 3 internal areas. So cut out that diagram for yourself. And now you realize that, because of this extreme length of the terminology list and the size of these diagrams I've provided, you're going to need to make a plan. So this is a quick and easy fix. Using 2 paper clips, especially if you have a wire-bound book like this one has been designed to be, you can just quickly clip in that extra diagram, and it will always be in a visible position for you when you're studying this part. We're going to keep up the color coding because there's no reason not to. So the forebrain is pretty much not just in the front as the name would suggest. It is everything that you would see as you are looking at the brain, including that internal part, which we will talk about again later. It's coming up in the terminology list. So linking up that forebrain color, and you will notice that none of these is mentioned in this subject matter. However, because there's a discussion about all the Broca's and Wernicke's areas, the basal ganglia, the amygdala, and all that stuff, they are often referred to by their location, either in forebrain or midbrain. So that is important to remember where those areas are. And the hindbrain, color coding that one, making sure to include that bit of the brainstem and the medulla, and then you've got your hindbrain, and that section is done. Now moving on, again, it's a recall instruction, so this is knowledge that you will have from grade 11. Because these 3 structures are areas that don't have specific structural identifiers, we could just use these stickers with a B for Broca's area and place that just on the edge there between your temporal and frontal lobe area. That is good enough an indicator for you about the location. Highlight it, color code it for yourself, and then we will just briefly put down a note about what that Broca's area does for you. Obviously being linked to that temporal lobe area, and on the left hand side of the brain, we will remember that it has a big job in the language processing. Specifically, you will see there in speech production. So being connected with the frontal lobe and voluntary movements, we can understand that how your mouth and tongue is going to form the words will be processed between the frontal and temporal. If your brain is going to be processing different kinds of muscle movements, you are going to produce different kinds of speech. The W in the back there for the Wernicke's area. And you'll notice how I'm lining them up in the notes. On the left hand side is the Broca's, on the right hand side is the Wernicke's, and that is how they are mostly going to be displayed in any diagrams that you will see or be given in the exam. So the front one is Broca's, the back one is Wernicke's. Now for this area, we'll see it is more about the understanding of what the language is telling you, so that verbal communication, understanding what's being said to you and what you are saying yourself. so processing that language. And here it's interesting that we see the writing part of it is also involved with the Wernicke's area. The third part is always very tricky because you'll find different representations of it depending on which texts you are looking at. But this Gershwin's territory really is an area that connects Broca's and Wernicke's and stretches up there into that parietal lobe. So it's definitely a territory description, which is obviously larger than an area. So for Gershwin's, it is important to remember that it kind of acts as a synonym for the inferior parietal lobe. Now, inferior would imply the bottom part of the parietal lobe, and you'll see there it is all to do with both your senses and motor, so movement stuff being involved. And because we've now seen that the parietal lobe is involved in all of the senses, including sight, you can see how it's also involved in visuomotor processing, so making your eyeballs go left, right, up or down, etc., together with the auditory processing working with Broca's and Wernicke's. Now, if there is going to be an issue with any of these areas and how they're communicating with each other, that is where we're going to come across the word aphasia. And I'm putting it in a bubble there because it is not a physical structure that we have to deal with. It is essentially a psychological condition, a disorder of the brain occurring in people often that have had strokes or brain damage from accidents. And it's going to affect their communication. So aphasia, that prefix, means without. So they're without one of the aspects of their language communication. So it can result in broken speech. That's a problem in the Broca's area. Or it can be a problem with them producing the correct words that they want to say. And that's a Wernicke's area issue. And you'll see it can affect their writing of language as well as their speaking. Now, the next terminology is part of the frontal lobe, so your primary motor cortex. Motor obviously implying that link to the voluntary movements. And what is important to put in your notes here with regards to that primary motor cortex is the fact that it's not so much about doing the motion, it is more about the direction of the motion. So if you want to reach for a glass in front of you, you are not going to reach behind you. Then finally we get to the last part of this diagram, which is the cerebellum. And I've run out of markers, so you make a plan, and I've just underlined that term clearly. Let's track again and see where we are in the syllabus. So we've done that recall, we have done that recognize, the primary motor cortex. We are now at the cerebellum and basal ganglia. Again, recognize implies prior knowledge. So we've learned previously that the cerebellum is very much involved with muscle control and your balance and your movement. Often with babies, if there is an issue, they will struggle to sit up, for example. Make your link to the primary motor cortex, and that diagram is done. Next up, we have the basal ganglia. Now that diagram is going to need to be added into your page, and because we've only used paper clips and not glue, you don't have to worry about ripping out or making any kind of weird things. You can just move your paper clips and attach that diagram with some glue, and all of it will be folded around, making it easier to access. Now, for this one, I'm using the purple again simply to imply that it is part of that forebrain. And you'll see that is where I'm going to add the basal ganglia terminology. That way, color coding it makes your brain remember what is going on and where all these parts are. And again, we'll see that the connection between that forebrain and the basal ganglia is control over all things that are voluntary. Do you want to move? Do you want to speak? Do you want to get angry at somebody? That kind of thing. Then we'll go to that emotion section. But before we get there, another term which is not officially on that syllabus list, which is the motor homunculus. Now, that is a really weird diagram if you're going to start checking it on Google Images, but it is very much linked to what is happening in the parietal lobe. So we refer to the motor homunculus as a topographical map. So essentially, where along the surface of the parietal lobe will the connection to specific sensory perceptions be? So where on the brain are you going to have control of experiencing what your foot is touching or what your eye is going to be seeing, etc. So it's very much linked to the specific parts of your brain and making that connection. So a simple version is what you can draw in for yourself there. That's obviously one of the sides of the brain. And in that little gap between the left and right hemispheres is where the foot information is going to be, curving around for the leg. And I've made the hand really, really large because it is a really large area that processes that touch sensitivity, followed by the eye, then whatever is smelt by the nose, and a larger area again for what is experienced by the mouth and the tongue. That brings us to the last few parts, recognizing again prior knowledge, bringing that in to the syllabus of the whole limbic system. Now, for the limbic system, which encompasses a lot of that area in the inside of the forebrain, we'll just indicate that by that light blue color. And obviously a system implies numerous different parts, so that is why it covers that whole area. So for this limbic system, we don't have a lot of space left on the page, but we don't need it, thankfully, because it is just a recognized prior knowledge kind of indicator and a cognition on the syllabus. So if you put a reminder there for yourself that your whole limbic system is linked to your behavior and your emotion, that is going to be good enough for you to remember, because then we can indicate that specific amygdala position. And that amygdala, remember, is the one that is really involved with you potentially losing your temper. So if you're experiencing that fight or flight situation and you decide that aggression is the best response, that amygdala, that's the basic location of it on a brain diagram. That is going to do what they describe as flipping your lid. So your amygdala, mark it there, and we'll put a reminder that it's all about the anxiety that you experience, the fear, and how you are going to respond to that. Leaving us with the area there on the frontal lobe, our prefrontal cortex. That just implies that you're looking at the in front of the front kind of location. So on the frontal lobe, pre meaning before, prefrontal cortex. So that is still going to be involved in your personality, your problem solving, and all of those different aspects that we've already covered in the frontal lobe. So again, just checking that we have covered everything, done, and it brings us to that last section, which were the last few terms on that terminology list. Now creating the summary. Obviously we have run out of space on this page, so grabbing an extra piece of lined paper would be handy. And you can create the summary using the subject matter, as I am basically doing now, or you can summarize it from what you have written on your own page already. Remembering that once you are done, you can bring back the color coding just to make it nice and visible. The aim of these summaries is pretty much to really be the crux of what you need. That is what you might go through the morning before the exam. Not that that's a good idea. Don't put that pressure on your brain, but some people prefer to do that. You can quickly, in that space that is left at the bottom of the page, glue in that summary. You can also use a piece of tape to make sure that it's not going anywhere, and obviously you will fold it up and away out of sight to use it again when you are preparing for those assessments. Don't underestimate the importance of these colors that you are using. You will find that very often they will help you to quickly find something on your pages, or your brain will remember, oh yes, it was the green thing, it was the blue thing. What was that blue thing linked to? Oh, excellent, it was linked to maybe the midbrain or the hindbrain, etc. And that's it. 2 of the 3 parts of this brain topic is done, so we can move on to the last part. Same process as before. Grab the terminology list, have a look at that last section. So in this case, they are all italicized. So you'll be starting at the synapse and running through neurotransmitter all the way to Alzheimer's. Again, you will notice that the specific terms synapse and neurotransmitter are not mentioned in the syllabus, but they are important things to remember from your prior learning, and they will repeatedly come up. If you have to explain neurotransmission, obviously you have to know what a synapse is, what it looks like, where it's going to be found. So communicate neurotransmission using a diagram. Then distinguish and compare and discuss, and you'll notice those 4 are much more complex cognitions, so they will most definitely be asked as direct questions in the exam. Grab those 2 relevant diagrams, space them out nicely to leave some area for writing above, and then you can start with neurotransmitter. However, you will notice I'm starting with neurotransmission as the process which involves those neurotransmitters, just to link in nicely with the syllabus. Reminding yourself that the neurotransmission is electrical impulses that gets relayed between those neurons. that is the transmission or the movement of those signals that we are interested in. Then we get to neurotransmitter, and I have colored in all of the little molecules that I know to be the neurotransmitters, just to show again visually where they're going to be found, and to make that connection that they are secreted by the neurons themselves. They will cross that synaptic gap and carry that message forward. Arrows are always important when we're talking about the directionality of these signals. And remember, it's going to be important for you to be specific that it goes from electrical in the neuron to chemical across the synapse, back to electrical in the other neuron. So that is then where your synapse or the synaptic gap is going to come in play in the terminology list. Now, you've noticed I've put in air quotes there. It's the connection, because we know there's no physical connection, but the chemicals that are released into the gap connect the two neurons with each other. So done. Be aware that you might be required to actually draw a process of neurotransmission, so potentially drawing a synaptic gap. Then we finally get to talk about new stuff, which is the types of neurotransmitters. So two categories, the excitatory and the inhibitory. Now their names pretty much tell you what they do. So the excitatory ones promote the creation or the making of an electrical signal. So they will stimulate the next neuron in the chain to restart that electrical zap, or the action potential, which is the fancy name for it. And it does that by forcing those positively charged sodiums to cross that membrane to depolarize that neuron. The inhibitory ones, on the other hand, will have the opposite effect. Now, please don't get confused or misunderstand. An inhibitory neurotransmitter is not necessarily a bad thing. All right? It does not necessarily inhibit your brain or your neurons from doing anything. It is simply how it's going to act on those electrical signals. So it stops an action potential, because now it promotes the negative charged chlorines to hyperpolarize that neuron. You don't want to be on an adrenaline high forever. You need things to stop. So that would be, for example, how an inhibitory neurotransmitter does something good for you. Now, the first ones that they want you to talk about in the syllabus is the glutamate and GABA. So for glutamate, it opens those channels because it obviously is going to try to promote the flowing through of those positive sodiums. So it makes for a stronger electrical impulse or electrical charge. Then the friend to glutamate, and notice how I'm grouping them under the category that applies to them. So glutamate is your most common one. It is involved in learning and memory, and its friend epinephrine, which is also commonly known as our adrenaline, that is obviously going to be involved in your flight, fight, or freeze responses. And it's useful during times of stress, when you're making a speech in front of the class or you're running away from something angry. And then also in the class of excitatory neurotransmitters is norepinephrine. Now, norepinephrine is not the opposite of epinephrine. All right, it is also involved in your fight or flight response. It increases your alertness. It increases your brain and body activeness. So your readiness to think about something or your readiness to run away. Then the last one that we can put under this side is the acetylcholine. Now here as well, when you start doing your own research, you'll notice that often it is referred to as an excitatory mediator, so kind of helping the other excitatory neurotransmitters. But both it and norepinephrine can also have certain inhibitory responses. So those two could be classified as both excitatory and inhibitory. The part for acetylcholine, which is important for our future knowledge about the learning and the memory and the REM sleep, as well as your attention, is the functions that we'll put in there. Now GABA, our friend gamma aminobutyric acid, you can just refer to it as GABA, also plays a big role in memory and in your cognitive functions. It is our main or first inhibitory type. That's why we put it at the top of the list. Then for dopamine. Dopamine is all about moving and motivating the movements. It's about experiencing pleasure. So if you're doing a hobby, do you enjoy it? It also is involved with your attention and memory. Then our last inhibitory friend, serotonin. This is definitely involved in our happiness. So a lot of people that are suffering from anxiety or depressions even do go onto serotonin meds to help with that. It reduces stress as well. So have we covered all the aspects of that in the syllabus? We've done their basic function, both physical and psychological. Yep. That leaves us with a discussion about Parkinson's and Alzheimer's. So for Parkinson's, these things are very familiar to us these days. Any damage to those neurotransmitters, specifically the dopamine and the dopamine receptors, is going to cause the problem, because dopamine helps you to do smooth movements. If that dopamine is not there, your movements are no longer going to be smooth. The treatment for that tends to be L-DOPA, or what we call levodopa. It is a precursor to dopamine, and that is in the medication simply to cross the blood-brain barrier. So you send the ingredients to your brain, and then your brain can make the dopamine itself. And the last one there on the list, Alzheimer's. Now we all know that Alzheimer's is our memory-related one. And the same as how we've made that connection with movement for Parkinson's, we will do the connection with memory when we are done with the Alzheimer's notes. So everywhere on your page where you've indicated memory is a function, that is the connection that you can make to Alzheimer's. And so the treatment for Alzheimer's is NMDA, which is N-methyl D-aspartate. It is a glutamate receptor, so it's going to help you pretty much get that glutamate to do what it needs to do. It will only take care of the symptoms. It will not remove that disorder from the brain. So done on the syllabus, everything is good to go. Just the summary. Again, you can use the syllabus document to create your summary or the notes on your page. Don't forget the color coding, which you can do for memory retention, and glue that into the bottom of the page. Use a bit of tape to secure it, make sure it doesn't get lost, and the entire brain topic is done. So this is about a third of Unit 3, which is finished. 3 pages only that you have to worry about when you are studying. Nice and short and sweet. You can fill in the page numbers. Check that everything is in place. It's not going to fall out. Thank you very much. I hope that helped with this first topic. See you in the next one.